and the monocytes, and of course the lymphocytes have sub-varieties. If you look at the lymphocytes, let's focus on those for a minute. About 80% of them in circulation are T lymphocytes. The 15% or so are B lymphocytes, and then 5% are called null. They're not apparently B or T. They actually they're sort of stem cells, if you will, that will give rise to B or T. There are two types of T cells. There are a whole bunch of types of these cells, but we're going to focus on just a couple of them. One is T killer cells, also called cytotoxic cells. These express a protein on their surface called CD8. The other type is a T helper cell. These have CD4 receptors on their surface, and they have separate functions. Now, T lymphocytes, B lymphocytes, these are all part of the immune system. One of the things that they're responsible for is to recognize things that don't belong to you. Cell surface markers that don't belong to you. Whether they be bacterial, whether they be viral, whether they be your kidney that's been transplanted into me, whatever. That's one of their goals, is to recognize foreign or non-self. Now, the cell surface marker that characterizes you, these are called the major histocompatibility complex, also called the human leukocyte antigen, which was the first one discovered. It was on a white blood cell, but it turns out all cells have these things, so now they're not called HLAs, they're called MHCs. That's the proper term, although HLA has kind of hung around, and you'll probably hear it referred to as, from time to time, but it's really the MHCs. What are the MHCs? When they do tissue typing in terms of organ donation, they're looking for closeness of the HLCs. The more foreign it is, the more likely you're going to recognize it as such, and you're going to reject it. My identical twin has the same MHCs as I do. We could transplant anything, and no one would, our bodies would never, I don't have an identical twin, I'm just giving an example.
I was like, you do?
I know what you, no, I saw the expression like, holy shit, there's two of you?
That's exactly what I thought.
No, but you see what I mean? Okay? That's why your brother, your sister, you share similarities in MHCs, but not so much your cousin. And certainly not so much the guy living next door, in the house next door to you. These are lymphocytes, and you see they don't have granules. They do have granules, as I said earlier, but they're just too small to really pick up. They've got an indented nucleus. It's not a lobed nucleus, it's just kind of indented. One more word about HLA, and then I'll forget it. I'll move away from it. Certain markers on cell surfaces predispose you to disease. Because, well, it can be a bunch of different reasons, but one reason they can predispose you to disease is the autoimmune nature of recognizing self. If I make antibodies to the beta cells of your pancreas, it will destroy them, and you will have diabetes. why would you make antibodies to your own beta cells? That's the basis of autoimmune disease. That's what Sjogren's is. It's you attacking yourself. The vasculature, it's a vasculitis. So the prevalence of certain HLAs, and the pathognomonic one is HLA-B27. If somebody says you have HLA-B27, the correct answer is ankylosing spondylitis. Ta-da. That's all I want to say about HLA. You're going to see the term hanging around. All right, now MHCs. There are two classes of MHCs. Class 2 MHCs are found only on B lymphocytes. Class 1 MHCs are on every other cell of your body. In this slide here, this is a B cell because we're talking about class 2 MHCs. This is a B cell, a B lymphocyte. What happens here is you've got, let me get the glare off here. You've got antigens circulating in the, let's say from bacteria, strep. The strep, once it enters your body, the strep dies, it breaks up. You've got millions of proteins or millions of sequences of amino acids from the strep that are different than your own amino acid sequences, right? They bind to receptors on B lymphocytes. These receptors internalize it and chop these fragments of strep protein up, into about 10 to 20 residues, meaning amino acids. They attach them to a newly formed class 2 MHC, and then they stick them in a vesicle that takes it to the surface where it fuses. And now you've got class 2 MHCs expressing the strep antigens. Pretty simple. This cell is now sensitized to strep-related proteins. The same thing happens to your own tissues. Your own tissues break down, right? You have normal apoptosis. Now this happened back when you were an embryo and a fetus. This happened a long time ago. Your own tissues started breaking down as you were developing. They bound to receptors, not only on B cells, but all cells of your body. Your muscle cells, your fat cells, your skin cells, everything. They got internalized, they got bound to an either class 1 MHC or a class 2. They went back to the surface, and now you have out here MHCs displaying self. My MHCs display Dan. Your MHCs express pieces of Meredith. That's why my MHC is different than yours. If I put my kidney in you, I have my expression on the surface, not your expression. It's pretty simple, right? I mean, it's a simple concept. It not only happens with pathological things, but it happens with your own tissue. That's how you recognize yourself. Your MHCs are displaying pieces of you. There are all sorts of markers on the cell surfaces. Here's the class 1, class 2 MHCs. You don't have them both together on the same cell. That ain't happening. MH2s are on B cells. B lymphocytes, MHC1s everywhere else. Okay? There's also another thing called a T cell receptor. We'll talk about in a second. And here we have our CD4 and CD8 cell surface markers. This is what it looks like when things bind. And I'm sorry this image is so dark, but you can read it over here. Here's an antigen-presenting cell. Let's say that's my kidney cell transplanted into you. It has my MHC attached, it has myself attached to my MHC. It will bind to the T cell receptor here. My T cell receptors will recognize this as non-self. And once it binds, the cell will either have a CD4 or a CD8 protein attached to it that will help anchor this relationship. That's how you recognize me as non-self. Now, if we look at this slide right here, you kind of follow me along here, because you don't want cells recognizing self, right? You don't want your lymphocytes recognizing self. Because remember, we're talking about CD4 positive or CD8 positive lymphocytes. That's what we're talking about here. CD4 or CD8 positive lymphocytes. Killers, right? We don't want to be doing that. So, back when you were a tiny little embryo, you had these thymus cells, the thymus, remember the thymus, the big thing sitting right here, it went away. You have these thymus cells that are displaying either class 1 MHCs or class 2 MHCs on their surface, displaying you, displaying self antigen.
I'm sorry, I don't follow this at all.
Okay, all right, so let me start this slide over. So the point we're going to get to here is lymphocytes, CD8 or CD4 lymphocytes, because that's your primary immune mechanism. But you don't want lymphocytes recognizing self. We only want lymphocytes in your circulation to recognize foreign subjects, right? This started back when you were a little bitty embryo and you had a thymus, and continued on till you were several years old. Your thymus cells had on their surface either class 1 or class 2 MHCs. Now, going back to this thing right here, you got to understand this. Right here, we've taken pieces of you. You know, we talked about apoptosis, how cells turn over normally, right? When they turn over, what are cells made of? Tons of proteins. They take chunks of those proteins and wind up getting bound to these receptors, internalized, and returned to the surface as markers for you. That's how you recognize yourself. All right? So, back when you were a little bitty kid, you had these thymus cells displaying pieces of you as either class 1 or class 2 MHCs. Now what we're going to do is we're going to take the immature T cell, and immature T cells are found in the bone marrow, they're found in lymph nodes, they're found in the spleen, they're found in tons of places, and you produce a billion of them a day. as an immature T cell, they contain a T cell receptor. If we go back to this slide right here, there's the T cell receptor. And as an immature cell, they contain both CD4 and CD8 cell surface receptors. If that T cell receptor binds to the thymic cells, if that T cell receptor binds to the thymic cell, either here or here, that means that lymphocyte has recognized self. We do not want it in the circulation. We kill it. That process is called clonal deletion. You prevent lymphocytes that recognize self from entering the circulation. If it doesn't bind here or here, we let it enter the circulation and it will either lose the CD4 or the CD8 receptor, and there you go. You have circulating T lymphocytes that do not recognize you, that are either CD4 positive or CD8 positive. What do you think autoimmune disease is? Autoimmune diseases is when those little son of bitches got into the circulation. They recognized you and started to attack the synovial membranes around your joints. That was rheumatoid arthritis. They attacked the parietal cells of your gut. Now you have pernicious anemia. They attack the beta cells of your pancreas. Now you have diabetes. They attack your thyroid. Now you have Hashimoto's thyroiditis. That's what autoimmune disease is.
What's the difference between that and clonal deletion then?
Clonal deletion is you got rid of them. You killed them. These are all clones of the same cell. You're just getting rid of the ones that recognize self. And I guarantee you, over the next 20 years, you're going to find that most disease is related to this, one way or another. And, if it's not infectious, it's related to this. Most diseases, when you say we don't know what causes it, I guarantee you it's going to be because of an autoimmune disorder. Here's a summary of our CD4 and our CD8 cells. CD4 positive T lymphocytes, they bind to class 2 MHCs. CD8 positive lymphocytes bind to class 1 MHCs. Where do you find class 2 MHCs? On B lymphocytes. What do B lymphocytes do? When they're activated, they turn into plasma cells that churn out immunoglobulin against some antigen. That's in a subsequent slide, so if you didn't get it, you'll get the second. If you want to turn on antibody production against an antigen, you've got to have your CD4 positive T lymphocytes to stimulate that. Without them, you don't mount an immunologic defense. And that's why people with HIV or AIDS die of opportunistic infections, because they can't mount an immunoglobulin defense, because their CD4 counts are too low. And this is why. This is humoral immunity. Remember, back in undergraduate, that they always call it humoral immunity. The reason it's humoral immunity is this. CD8 cells, these guys, what they do is they are looking for non-self class 1 MHCs, like on that kidney cell. They're going to bind to it, and then they take this protein called perforin. It's like a toilet paper tube. The little cardboard toilet paper tube, it's a protein in a tube, and they stick it into the cell membrane of my kidney that I transplanted into you. Now you got an opening, all the stuff goes out of the cell and it dies. That's why it's called a killer cell. Okay? See, it's easy, it makes sense.
Oh, yeah.
Lymphocyte stages, lymphoblast, prolymphocytes, and lymphocytes. Lymphoblastic leukemia is not good. It's an undifferentiated leukemia of lymphocytes. By the way, just to give you a heads up, I'll give you a heads up. There are 4 basic types of leukemia: acute and chronic, lymphocytic leukemia, and acute and chronic myelogenous leukemia. You would say it again?
Yes.
Yeah, it's acute and chronic lymphocytic leukemia or acute and chronic myelogenous, M-Y-E-L-O, genus leukemia. ALL or CLL. The malignancy is a lymphocyte. In AML, acute myelogenous leukemia, or chronic myelogenous leukemia, CML, the malignant cell type is a granulocyte. So when they start talking to you about the different leukemias this week, that's basically what they're talking about. It's either coming from the lymphocyte cell line or the granulocyte cell line. It's that simple. Okay, monocytes, as I mentioned to you before, they're not mature cells in the circulation. They have to crawl through and get extravascular to become macrophages. What they do is they eat everything. They'll eat everything in their path. They don't last very long once they leave the circulation, but they leave in droves. Okay? Here's a monocyte here. The thing about a monocyte is that the nucleus is irregular shaped. It's kind of the hallmark of a monocyte. A lymphocyte has a nice round nucleus. Monocytes is irregular. Plasma cells, I said that plasma cells are activated B lymphocytes. You'll find them they're not in circulation, they are in the connective tissues. They are in places where you have exposure to the outside world, like in the GI tract around your anus, around your oral cavity, around your nasal cavity, in the connective tissues, in those spaces. Or, your eye, your ears, different places like that. They have a clock-based nucleus because of the clumps of chromatin around the edge of the nucleus. They're really obvious when you look at tissue, subcutaneous tissue, when you find them. All right, so here's the thing. IgEs bind to mast cells. These guys secrete IgEs in response to, when they're activated, they dump out IgEs. There are like 5 different types of immunoglobulins: IgA, IgG, IgM, IgE, and so on, right? These guys dump out IgGs that bind to mast cells. When they do that, it causes the mast cells to degranulate, dumping these things into the surrounding tissues as chemoattractants, as they alter the vascularity, they make it more permeable. So here's it laid out for you. There's a plasma cell, there's a mast cell, there's a degranulation. So when you take certain drugs to treat asthma or allergies, you use a mast cell stabilizer that blocks this reaction here. It blocks the relationship between plasma cells and mast cells. And that's it. I can promise you one thing, tomorrow morning at 8 o'clock, the CBC, it will be easy. So, have a good night, get some rest. Those who are going to Good Shepherd, enjoy yourself. We'll see you tomorrow morning. Any questions?